Although microbes such as bacteria, protozoans, viruses and viroids are found everywhere, they are quite at home inside your mother's kitchen. And why not? Since microbes help us prepare various useful household products such as curd, bread, idli dosa, dhokla, to name just a few. For example, have you ever wondered how milk, when left overnight with a little curd in a warm place, turns into curd by morning? The little curd added to the milk initially acts as an inoculum or starter that has useful bacteria called lactobacillus, also known as lactic acid bacteria or LAB. LAB, which is present in millions inside the inoculum, multiplies at a suitable temperature and converts milk sugar lactose into lactic acid. The lactic acid in turn makes the milk thicker and partly digests the milk proteins, resulting in the formation of curd. LAB improves the nutritional quality as the curd now has increased quantities of vitamin B12. After the curd is prepared, it is stored in a cool place so that the LAB doesn't make it excessively sour by producing lactic acid. Another benefit of LAB is to check the presence of harmful bacteria inside our stomach. In addition, some bacteria help in curing of tea, cheese and tobacco. Curing is the process that determines the flavor of tea, cheese and tobacco based on the degree of fermentation and putrefaction they have undergone by bacteria. Different varieties of cheese are distinguished by their characteristic texture, flavor and taste, all of which depend on the choice of microbe used. For example, Swiss cheese has large holes because bacteria Propionibacterium sharmani produce a large amount of carbon dioxide. Roquefort cheese, on the other hand, acquires its characteristic flavor due to a specific fungus called Penicillium roqueforti that is used to ripen it. Microbes help in making of other traditional food like bread, idli dosa, dhokla and khaman by a common process of fermentation. To make bread, baker's yeast or Saccharomyces cerevisiae is added to uncooked dough. The dough rises due to exhalation of carbon dioxide by the yeast. Now, when the bread is baked, the carbon dioxide escapes to leave a porous and light bread. Yeast helps in the preparation of idlis and dosas in a similar manner. A mixture of ground rice and dal is allowed to stand overnight and ferment. This mixture rises and sours due to the growth of yeast cells, which lends the sour flavor in the idli and dosa. In case of dhokla and khaman, the batter is prepared in a similar way with Bengal gram dal and left overnight to be fermented by microbes. Yeast also helps in wine and beer preparation by fermenting the sugars present in fruit juices or barley to produce alcohol and carbon dioxide. This is the same principle that is used in the production of toddy, a traditional drink in southern India, made by fermenting the sap of the palm tree. Similarly, aerobic bacteria called Acetobacter converts ethyl alcohol to produce acetic acid or vinegar. This explains why the word vinegar means sour wine. Microbes are also used to ferment fish, soya bean and bamboo shoots to prepare different foods. Therefore, several of our traditional food and drinks are derived from various microbes every day. Microbes have proved to be useful inside our kitchen by helping us prepare various food items such as bread, curd, dosa, wine, and toddy. Naturally, the same underlying principle of fermentation that is used to prepare these products is also applied on a large scale in various industries. In industries, microbes are grown in large vessels called fermenters that are used to make beverages like wine, beer, whiskey, brandy, and rum on a large scale. One such microbe is Saccharomyces cerevisiae, that is known as baker's yeast due to its use in bread making. It also acts as brewer's yeast by fermenting different malted cereals and fruit juices to produce ethanol. So, depending on the type of beverage used for fermentation and the type of processing such as distillation, different alcoholic drinks are prepared. For example, wine and beer, which are low in alcoholic content, are prepared by fermentation of sugar or starch products from plants, and their production does not involve distillation. On the other hand, whiskey, brandy and rum, which are high in alcoholic content, are produced by distillation of fermented malted barley, grape juice, and molasses respectively. Other than alcoholic beverages, microbes help in the industrial production of antibiotics, chemicals, enzymes, and other bioactive molecules. Antibiotics are chemicals produced by some microbes that can kill or check other harmful disease-causing microbes. In Greek, the word antibiotic means against life, that is, against the life of harmful disease-causing microbes. Penicillin is the first antibiotic that was discovered by Alexander Fleming in 1928 by serendipity. While working on bacteria staphylococci, Alexander Fleming discovered that this bacterium did not grow around a particular mold called Penicillium notatum on unwashed culture plate. He realized that bacteria did not grow around the mold Penicillium notatum as it released a chemical. He called this chemical penicillin after the mold. Though Alexander Fleming discovered penicillin, it was Ernest Chain and Howard Florey who applied the curative effect of penicillin against various infectious diseases. During World War II, penicillin was effectively used to treat several soldiers. For this contribution, both Chain and Florey were awarded the 1945 Nobel Prize in Physiology and Medicine along with Sir Alexander Fleming. After penicillin, other new antibiotics such as streptomycin that was used to treat tuberculosis were discovered. Streptomycin, along with other antibiotics like actinomycin, streptothricin, and neomycin, were all produced by different species of bacterial genera Streptomyces. Since then, more antibiotics have been discovered that have greatly helped save lives around the world by curing various infectious diseases such as diphtheria, leprosy, plague, and whooping cough. Besides alcohol and antibiotics, microbes also produce chemicals such as organic acids. A fungus, Aspergillus niger, helps produce citric acid, which is very useful for the soft drinks industry. Similarly, microbes such as Acetobacter, Clostridium botulism, and Lactobacillus or Lab produce acetic acid, butyric acid, and lactic acid respectively. Like acid, the production of enzymes by microbes has proved to be very useful for industries. For example, enzymes such as lipases are used in detergents for their ability to remove oily stains. Industrial lipase is produced from fungi such as Candida rugosa and Aspergillus niger. Likewise, pectinases and proteases produced by microbes such as Aspergillus niger are used in bottled fruit juices for clarification. This is why bottled fruit juices bought from the market are so much clearer than those prepared at home. Another enzyme, streptokinase, which is produced by bacteria Streptococcus, is genetically engineered to act as a clot buster. It is used to remove clots from blood vessels in patients suffering from myocardial infarction, commonly known as a heart attack. Furthermore, statins produced by yeast Monascus purpureus are used as blood cholesterol-lowering agents. They work by competitively inhibiting the enzyme that produces cholesterol in the liver. Cyclosporin A is another useful microbial offering by fungus Trichoderma polysporum. It is administered as an immunosuppressant to organ transplant patients so that their immune system doesn't reject the transplant. Lastly, bacteria and fungi are also used in microbiological cultures, where they are grown on nutritive solutions and allowed to colonize so that they can be studied. Therefore, microbes such as bacteria and fungi play an important role in the production of products such as alcoholic beverages, antibiotics, acids, enzymes, and other chemicals on an industrial level. Every city or village produces millions of tons of wastewater, spent water from residences that carry body wastes, excreta, food remains, and laundry wastes, to name a few. Similarly, industries spew commercial wastes through wastewater. All such wastewater is called sewage. Have you ever wondered where all this sewage produced by us goes? It certainly cannot be just deposited as it is into rivers or the sea. Sewage is first treated by microbes that are naturally present inside it before it can be safely disposed of in rivers and streams to avoid polluting these water bodies. That is why the Ministry of Environment and Forests has produced the construction of a large number of sewage treatment plants under the Ganga Action Plan and Yamuna Action Plan to save major rivers like the Ganga and Yamuna. In India, microbes are already treating gallons of wastewater inside several sewage plants around the country every day. Let's take a look at how microbes help in sewage treatment. Microbial treatment of sewage usually takes place in two stages: primary and secondary treatment. In the primary stage, the objective is to remove coarse solids and other large and small particles found in sewage. The first step of primary treatment involves filtration, which is followed by grit removal inside a primary settling tank. All the floating debris is removed by sequential filtration, whereas the grit, such as coarse soil or pebbles, is removed via sedimentation. During sedimentation, most organic and inorganic solids settle, while floating materials like grease and plastic rise to the surface and are skimmed off. The settled solids are called primary sludge, while the floating material comprises primary effluents. Primary effluents are now transferred from the primary settling tank to large aeration tanks for secondary treatment, also known as biological treatment. Secondary treatment aims to reduce organic matter in the effluents. In the aeration tanks, the effluents are constantly agitated mechanically and oxygen is pumped into them. As a result, aerobic bacteria naturally present in the effluents grow into flocks, which are mesh-like structures formed by groups of bacteria in association with fungal hyphae or filaments. In the presence of oxygen, these growing aerobic microbes consume the organic matter present in the effluents. This secondary treatment reduces the BOD, or biochemical oxygen demand of water. BOD is an important measure of water quality. It refers to the amount of oxygen needed by bacteria and other microbes to oxidize all the organic matter in 1 liter of water. The BOD of drinking water should be less than 0.5. However, the BOD of raw sewage can be as high as 600 milligrams per liter. That is, the greater the BOD of wastewater, the more polluted it is. Also, it is an indicator of the amount of organic matter present in water. Secondary treatment is continued till BOD decreases considerably. Once this is accomplished, the effluents are transferred to another aeration tank where the flocks of bacteria are gradually allowed to sediment. This sedimented part of sewage is known as activated sludge, which is pumped into large tanks called anaerobic sludge digesters. Here, anaerobic bacteria digest the bacteria and fungi present in the activated sludge and produce a mixture of gases such as methane, hydrogen sulfide, and carbon dioxide. This mixture of gases is also called biogas and is used as fuel since it is inflammable. Methane is the main gas produced from sewage treatment and also serves as fuel. It is used to produce heat in homes and factories. The sludge left after digestion of both aerobic and anaerobic bacteria is used as manure and is called activated sludge. The effluents that now remain from the secondary treatment plant are now fit to be released into natural water bodies like rivers and streams. However, a small part of the activated sludge is pumped back into the aeration tank to serve as inoculums for subsequent treatments. Though the amount of sewage produced increases every year with the population increase and rapid urbanization, microbial treatment of sewage remains the most effective method to date. For thousands of years, since the time of ancient Egypt, yeast has been used for baking and fermentation. With time, it was observed that dough rose during the bread-making process due to the exhalation of carbon dioxide by yeast. When bread is baked, the carbon dioxide escapes, leaving a porous and light bread. Similarly, during fermentation, to prepare beverages and cheese, the main gas that yeasts produce is carbon dioxide. It is therefore evident that microbes produce gases during their growth and metabolism. Even so, different microbes produce different gases depending on the organic substrates they utilize. For example, anaerobic bacteria that grow on organic matter such as cellulose